Number 3. Leeches are parasites that feed by sucking blood. When they bite, they secrete saliva into the wound. The saliva contains a globular protein called hyaridin. Explain why this protein is soluble in water. Right. So you're sitting in an exam and you read this question, and there's a really high chance that you've never come across this globular protein, unless you're really interested in leeches. And there's a slight panic. And Edexcel does this. They will always give you a scenario or a topic that you've never heard of, but they will never ask you a question about something that you've never read about or that's not part of specification. So all these questions they ask you will never be about something that you don't know. You have read this in your A-level textbook and it's in the specification, but the background to it you will have certainly almost never heard of. And the thing is they try to make you sort of have a synoptic link where you can link them, but actually the question is much easier. So if we take a look at this question, explain why this protein is soluble in water, if we rephrase it, well, it's actually explain why a protein is soluble in water. Now that you know, you could answer that. And so that's how the questions are phrased, and that's the type of like background that you get given. But it's nothing that you can't give an answer to. And so why is this protein soluble in water? Well, why is a protein soluble in water? You know this. So let's go through it. So the first thing is that in order for a protein to be soluble in water, it must have hydrophilic groups that are on the outside. And so its hydrophobic groups will be on the inside. And so hydrophilic groups are on the outside of the protein. Now, what's another reason that you know that has properties that are associated with making a protein soluble in water? Well, proteins are made of amino acids. Amino acids all have the same structure. However, they differentiate in terms of an R group, and the R group is what makes the amino acid polar or ionic, and so that will give it the ability to be soluble in water. And so therefore, the exposed R groups are polar. And so if they're polar, it gives them a specific property in terms of they can make hydrogen bonds with water. Whenever you hear the word polar and you hear the word water, the answer is always going to be hydrogen bonds. Because when a polar substance comes into contact with water and it has a hydrogen atom that's connected to an oxygen or a nitrogen or a sulfur, they can form hydrogen bonds. And so the answer here is they form hydrogen bonds with water. And so that's your 3 marks. However, you can get another mark if you didn't state any, if you missed out one of these, by saying a property of water that allows the water to be mixed in with polar substances, and that's because water is polar. Water is a polar solvent. And so those are all the available marks for this question. Once again, these are the answers as everything that you should know. So, hyaridin is an inhibitor that forms hydrogen bonds with an enzyme. Explain how hyaridin would inhibit the activity of the enzyme. So once again, you would have never come across this protein, and so you wouldn't know how it inhibits an enzyme, but you do know how an inhibitor will inhibit an enzyme. And that's what this question is asking you. So how would an inhibitor inhibit the activity of an enzyme? That you do know. And so, what would inhibit the activity of an enzyme? Well, the first thing is that the inhibitor would change the structure of the enzyme. And so here we have to link it back to the question. Pyridine would change the structure of the enzyme. That's the first thing we know that causes inhibition of an enzyme. And now, because it says explain, whenever they say explain, it means say why. Describe means just state, whereas explain means you always have to give an why. Why does that happen? And so, well, we said it changes the structure of the enzyme. Well, how? Well, we know that there's a very important site on the enzyme that if that site is affected, the whole activity is affected, and that's the active site. And so we get changes in the active site of the enzyme. Now, so what if we have changes in the active site of the enzyme? You always have to remember, another good tip is that the person that's reading your answer knows nothing about biology. And so you have to explain every little detail. You have to explain every little thing. So we know that changes in the active site of the enzyme will mean that the enzyme won't either work or work as efficiently as before. But the reader does not know that. And so you've just told them that you get changes in the active site of the enzyme. That means nothing to them. So now you need to explain why changes in the active site of the enzyme is a big deal. Well, we know this because it would prevent the substrate from binding. That's why it's a big deal. Will stop the substrate from binding, and so therefore you will no longer have any reactions occurring. And so a good tip to always have in the back of your mind is that explain everything. Whenever there's an explain question, assume that the reader knows nothing, and so you have to explain every little thing to them. Whenever you write a sentence, say, why is this important? Why? Always explain it. And that's it.